Metastatic breast cancer is when cancer cells spread from the primary site to other sites in the body. So I think most people realise that you've got a kidney, you've got liver, you've got brain, and all of these tissues stay in the one place. Your liver stays together, the kidney is a separate organ, doesn't move away anywhere else, the muscle's separate, and all the parts of your body are maintained in different compartments. But what happens in a cancer is cells from one compartment decide to move to another place. And so that's called metastasis or movement. They detach themselves from the primary tumour and they spread around the body. I mean, of course, the exception is blood, where blood is a compartment that does move, but most of the solid tissues are fixed and they don't move. And so when a metastatic cancer develops, it means that the cancer is starting to move around the body. And you can still have a cancer that's not moving. That's called in situ. And so it's much easier to treat that because the cancer's in the original position with the rest of the tissue. You can treat that quite readily by radiotherapy or surgery. But once the cells start moving around the body, well, that creates a lot of problems because those cells get mixed up with the normal cells of other tissues. Well, they can spread through the lymph system or they can spread through the blood, but essentially they have to digest their way out of the original site, which is the mammary gland, and they get into the blood or into the lymph, and they can spread that way to other parts like the bone or the liver. It's a matter of those cells becoming detached and not so strongly adherent to each other, and they become separate, no longer a single clump, although they can migrate as clumps. They move into the blood and then they can recolonise somewhere else. Epithelial to mesenchymal transition is a way that we think of cancer. We have to understand what happens to cancer cells and why a normal cell suddenly becomes a cancer cell, or not suddenly, but over time. And because the cells are stuck together originally in the primary tissue, they don't move, and then they suddenly change, we have to come up with a model for that. And so what we think is that during cancer, the cells change their behaviour, and instead of being epithelial, they become mesenchymal and migratory. And a mesenchymal cell is a cell that's a separate cell. They don't adhere together like epithelial cells. One of the key concepts in that is that cancer is not actually an abnormal process, but it's a process that's inappropriate in an adult, because during development, as you probably know, you've got a ball of cells. You start off with an embryo and it becomes a massive cells, and then those cells start to differentiate. They start to migrate to form the peripheral nervous system, and so the cells that are migrating go from being epithelial to becoming mesenchymal. So we think that this epithelial to mesenchyme transition is happening in cancer inappropriately. So it's appropriate for this to happen in development because you need the organs, the peripheral nervous system and other things to develop, but it's not appropriate in an adult. So somehow the cells are being given a switch which converts them into being motile, and that's really when they become metastatic. Well, I wasn't working on cancer at all. I did my PhD on zinc, and when I came to Deakin, there was no lab there, and I didn't do any research for some years. But I had a lot of inquiries from students. And one student said to me, I want to work on breast cancer. And I thought, well, I don't know a lot about it, but maybe we'll get some breast cancer cells as a starting point. So I just contacted what was the Ludwig Institute, and I came across a scientist who was very pleased to give me some breast cancer cells. So my students started growing these, and at that time we had almost no facilities. There wasn't even a microscope. So we couldn't even see the cells. We just had to stain them. It was really basic. But she did some interesting work staining those cells. And we found that we could develop a culture that was able to, over a period of 10 years, we found that these breast cancer cells could be made to switch on and off from the epithelial to the mesenchyme and back and forward. And so that's how this whole concept came. There was even an epithelial to mesenchymal transition research group formed at a conference. So it's been quite a big concept in cancer research. When oncologists, that is cancer doctors, are seeing patients, they often take samples for analysis because they want to understand more about the cancer. So they might take a tissue sample, or they can take blood samples. So the tissue samples will contain maybe the cancer tissue, and the blood will contain its circulating tumour cells, which are the tumour cells that are probably maybe going to start a secondary site somewhere else. And so in some cases in a lot of cases we work with doctors, and they provide us with samples that we can analyse. And so from that we can either look at the tissues, or else if we have live tissue, we can grow cells from it and use those for doing laboratory studies. Well, the cancer cells, initially they're quite hard to culture. Most cells in the body will divide for 20 replications, and then they'll become old and senescent and die. So that's a normal thing. So the cells in the body are like, if you take them away from the body, they behave in the same way as the whole person does. Like a person has got a limited lifespan of about 100 years, a bit less maybe. And their cells, if you take them, reflect that. So if you take cells from a newborn baby, then those cells will divide for a long time. But if you take cells from an old person, those cells won't last very long. So we've devised all sorts of techniques for getting them to live indefinitely, and those include infecting them with inactive viruses that can switch on their growth genes so we can keep using them. They maintain their same characteristics. Human cells are quite hard to work with compared to bacterial cells. So we can grow in the lab cells ranging from bacteria to yeast to fungi to plants to animals to humans. Now I don't do animal experiments because I don't like doing things to animals. So I stick to humans, either human tissues or human cells, because they give you their consent or otherwise. So the hardest thing about growing human cells is that they're complex, and they have to be grown with special nutrients in a special environment with a certain amount of carbon dioxide, a certain amount of oxygen at 37 degrees. You've got to have growth factors, you've got to have amino acids. It's quite complex. Whereas with some of the more, single cell organisms like bacteria, you can grow them in much simpler media, and they'll grow at a range of temperatures. And it doesn't matter if you accidentally leave them out overnight or accidentally heat them up a bit. But human cells are you have to keep them within a very narrow range of conditions.